This is similar to what Dr. Asri already introduced in the morning about we have a lot of sequence right now, but just so you know that 10 years ago, if you see in the middle of the slide here, we thought that at that time we are in the middle of the sea of information, right? We are drowning because at that time, people think about the data generated within 48 hours. At that time, we are in the era of the second generation sequencing technology. So we generated about 2 gigabytes per 48 hours. At that time, we think that's a lot. And then now, 10 years later, now one machine, one run can generate up to 8 terabytes or 8,000 gigabytes in 48 hours. So with this, a lot of information flowing in the across the globe. You can see in the middle, there's an estimation here. I got this statistic from the public repository that people deposit the genome. The name is the GenBank. If you count the viral genome, we are around 4 million. We have around 4 million genomes right now to analyze. So with this rate of the generation of the data, I think in the future we have a lot of data coming, and the goal is to keep up with this, and we need the tools that can help us easily generate some or some analyze some and get some meaningful meaning from this kind of data. And you can see there's an estimation from the paper. At that time, they estimate that the genomics stuff alone, the data generated in genomics field will, will be more than the, all the data from YouTube and all the Twitters or even NASA. So we need something. We need something to help us to deal with this large amount of data. And you can see if we can achieve that goal, we can use this data, in this case, genomic data, to analyze for example here. It either can be the microbial community and diseases. You can analyze for genetic markers for diseases, and also the most important thing right now, because we are in the pandemic now, that we can do biosurveillance, that you can see now is in action, that you can track, for example, the COVID-19, right? The SARS-CoV-2 virus. There are different strains of them, different variants. So you can see if you can analyze this data very fast, you can see where do they go, and which variants very common in some area or some population. So you can design the policy accordingly. So this is the way that we can use the genomic data in order to do the biosurveillance here. And this is the same thing. So my work is all about the computational biology, right? We do the computational work. So it is important to understand that actually the things that we're working on is the information in biological system. So everyone knows that DNA makes RNA makes protein, right? But this is an example. It's called central dogma, right? So, but actually, there's this one represents the information flow in the biological system. You have different layer of information here that you can make use of that. You can find the association. You can do many things with that. For example, in DNA level, you can compare DNA level. In the RNA, you can measure the expression of the gene you interest, and then at the end, in protein level, you can analyze for the similarity of the protein functional domain, right? What do they do, and what kind of unit of function that lies in the protein sequence, right? So all of this is the biological system information in flowing in the biological system. So just so you know that if you're doing the computational biology, you need to get familiar with different layers, and sometimes you need to combine them to find something meaningful. And go back to this slide again. So as I shown that and informed that there's a lot of sequence right now, and there's a lot of to come in the future. And this just only one dimension that I just have shown. This is just only DNA level, right? You have a genome sequence. But then it means that you have, once you have one level of the genomic of this biological information, you can turn it out into we can you can get the other information. Like for this example, like for example, one genome you can have many you can have a lot of potential protein sequences, right? So it means that if you have 4 million in this case in the database, meaning that you have a billion of proteins you can extract from those genomes. So so this is the reason why we need to build a lot of bioinformatic tools to cope with this kind of data, because we have a lot and the time is very expensive, right? So you don't want to stuck in some some analysis and then spend like a month on them and then just at the end just fail. So that's why this is the reason why we need to have some tool or the group of tools that help us to do in this case comparative genomics, right? We compare genome. We want to see what is in common, what is different among all genomes. So this is the first example. I think this one published in 2013 by Dr. Accessory Group when he was in Denmark. At that time, it's called CMG Bio2 or Comparative Genomics Bio2. And this is the workbench for basic comparative genomics. Meaning that you can use this tool to analyze any genome. Sorry. And you don't really need to have a very good skill in programming or something, because most of the time in academic the program will be delivered in the source code, right? You need to like run it by yourself, or you need to have at least some knowledge in programming to just fine tune them. And then there's a lot of the dependencies that you need to install in order to make those tools work, right? Not just only that tool, but just sometimes you need to install a lot of stuff and compile the program. So at that time, that's why they decide to deliver this tool in the virtual box or the virtual environment. Like you have either Windows or Mac machine, you can just install the virtual machine of the Linux or the Ubuntu in your machine, and then you just you can just use these tools at that time. So I think that is the way that you can deliver the software to someone who not that good at the programming skills. So you just build everything in one package and ship it to them and make sure everything works, right? And at that time it works well, but remember this is almost 10 years ago. And this is the plot or the features that you can compare using the CMG Bio tools. So and I think during the workshop you will I think Dr. Asri and our workshop will guide you through each of them. So what is that, and then how can you make them, and what will them tell you. So start from 16S, Blastomastic, all of this is the way the tools that help us to find the common or different in the genome that we are interested. And another problem that we have seen in the CMG Bio tool at that time, almost 10 years ago, is that you can see, even though you can, we deliver the virtual box, right, which is the virtual environment to the user. So meaning that user can just install the virtual box regardless of the system they are on. Like either they can be on Windows or they can be in the Mac, right? You just install the Linux in your system using VirtualBox. But then again, it's still the bunch of the tools packed together. So the user still need to dealing with the file formats and prepare everything in order to run. You can see from step one. Step one is that is it architecture at that time. You have the step one, and you need to get the genome first, right? And then the most problem part is the step two. So each of the tools here in the step three analysis, it require different input, right? The program, you can see here, there's a lot of program that can help you generate a plot in step 3. And each of them need different types of the data to feed in, right, to generate the plot. So we left the task to the user. You need to get the genome, then you need to run the program to generate the data that the program need in order to make the plot. So this is the most confusing part after you install the program, right? So you need to use that. You need to use a lot of the script to generate the data that fit with the tools that we have. So that's why we go back, and then, you know, this is 10 years after that, we think about how can we go to the sustained future, right? It's this one might confuse some the user. So how can we deal with this? And also another problem is that the way that we deliver the program is the virtual box, which require a lot of resource. As you can see here, this is the design of the virtual box. Of course, you need to have the machine, right? Everyone need to have the infrastructure. This is your machine. Then you need to install the virtual box, which is the tool that help you to make the virtual environment in your computer. You can see here, each of the virtual box need their own space and resource, right? Meaning that one resource cannot share with other resource. If you have 2 virtual machine in your computer, it would take 2 space, 2 slot here, take in your machine. So that's not the good design, right? I mean, we think that this one probably not make the good design for the software. For example, if you machine has the limited space in the hard disk, you might run out of the space, right? Because a 2 virtual machine cannot share their space, right? Or sometimes you can share, but it involves a lot of the complicated step in order to make it work. So that's why we moved to the new design. Oh. Okay. Okay. All right. So okay. Cannot see my okay. We have in the chat box. Okay. Sorry for that. Kind of the mouse is lost. So that's why we go back and think about how can we improve this. And then at the end we decided to go to the new the way of the shipping the program, that we decide to ship in the R package instead. So you can see at the bottom on the right hand side, the architecture of the software is much simpler compared to the one that we deliver in virtual machine. You can see you have just only in your infrastructure. It either can be your laptop or sometimes it's a server. You have operating system, right? And then you just install R and RStudio. Then you just install our tool, and that's all you need. And this design, it can take all the resource in your machine. So we eliminate the limit from the virtual machine, that they will not share any resource to each other. And also it's a lightweight design. So I think this way we can still do the computational part in your local machine, right? Without any take any resources, a lot of resources. So this is just the cartoon tell you that, okay, this is a SIMGBio tool 10 years ago. You can see just like the remote control at that time, it's super analog, right? We left the user like, okay, you need to make this kind of file format in order to feed in the program to generate the plot. Now you have the RBio tools, the much simpler. You can see less button, and it will reduce a lot of your work. for doing the comparative genomics. And this is the workflow. So this is the latest version of the RBio tool. So it requires you. You can see we cover, since we need to cover all the function that CMG Bio tool has, and then we want to transform it into RBio tools. That's why we need to rewrote most of the code again, and then we have some. Oops, I have the chat here. Chat box. Oh, okay. Sorry. Yeah. So in this case, we cover most of the function from CMG BioTool, and we convert to RBio. Just the last step that we are working on is that the genome atlas, right? This one, the last one that we haven't covered yet, but most of the function it works, and we hope that we can finish them and deliver the paper soon. And now we are preparing the manuscript for this too. And this is the last one that we're working on, the genome atlas. So this one allows you to compare, to see the property of the genome that you are interested. For example, this is the atlas of the Escherichia coli K12. And you can see this is the circular DNA, right? So you can see start from inside most, you have a lot of and the color code is on the right hand side. You can see from these genomes, you can immediately see where is the RNA, right? You can see the blue part within this plot. It would, sorry, it tells you the basic property that you can then see here from the innermost. See, you can see RNA directly within where it's in the genome, right? You can see here there's a blue part, which indicate the RNA, including 16S RNA. And then you can see the red and the blue here, which is the coding sequence or the potential, the place that you can, that can be the protein within this genome. And also each of the circle here represent the genomic property of this genome, like a GC skew, the position preference, and also stacking energy of this genome. Basically, it will tell you what is the basic property of the genome that you are interested here. Start from the first of the genome sequence here, right, from 0 to 45, 4 million, 4.5 million base pair here along this DNA sequence here. Where is the protein? Where is the RNA? So we are working on this. All right. Okay, so since we moved to the RBio tool instead of the virtual machine, so this is the requirements that you need to install in order to use our software here. You need to have the R programming language in your machine, right? So this is the link. So I will provide the link. Don't worry about that, but this is just to give you an overview what is the requirement for the new design of our tools. You need to have the R programming language, right? You need to install it. There's a link, and you need to have the R Studio or the IDE for developing the R code in your machine. So you can see in the right hand side, this is the design of the GUI of the R Studio. You can see there's 4 different panes here. So you can do the code. The first one is the code editor. You just like type any code that you want to use in our package, and then when you hit run it, the result of the message generated from the code will appear in the second pane, which is our console in the bottom left. And then you can always check what is the environment that you are on, and also the variables that you have on the third panes here. You can see on the bottom right, from the up right here in the third is a workspace and history, and the last one is the fourth plane. In the bottom right is the plot and the files. After you run it, it will generate a plot and files that from the script. So this one, it means that you can see everything in one place. In this case, we choose RStudio that help you to run the R code in your machine, so you can see what you run and then what you get immediately in on the right hand side in the plot. So this is very easy, rather than it's better, much better than the previous one, SimGBio2, that you need to generate one by one in the dark screen of command line dark screen, and you just uh, run the code and you need to open the files, the folder to see the results, right? So this one you just run and you can see immediately. So it's much easier for someone who don't really have the background in the programming. And the third one, the requirement is that you need to have the C compiler in your machine. This one will build our package in the RBio tools in your machine. So for those who use PC, you need to have the R tools. This is the 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 C compiler provided by the R group. So you need to install it. And for Macs, you need Xcode, right, installation. I think our assistant can help you for this. And at the end, you need the RBio tools. Right now, we have the version is 0.4. So this is the latest, and you can download from the following link. So don't worry, I will provide this link for you. So this is just tell you about the requirements. And yeah, this is the step for the RBio tool installation. You need to start to check in your machine that your machine support this, and install the C compiler first. For Mac people, you need Xcode, and for Windows, you need R tools. And after that, if you finish the install for the requirements, you can begin installing our RB dependency first. And in the right hand side, there's a bunch of the package that you need to install before you can use RBio tools. And then you can, after that, after you install all these dependencies, you need to install the protocol, which is the gene finder tools, and the RNA, which is the RNA finder. And at the end, you can install RBio tools. It seems very complicated, but I will have the demo to tell you that this is not the pain. This is just something that you need to have it before, and our environment will help you get all of this dependency installed. And this is the plot that you can, this is the feature of RBio tools. We try to very hard to make it similar to what we have in SimGBio tools, even though it requires writing the bunch of new code. For example, this is the 16S RNA, right? This is one of the things in comparative genomics that you need to know, like how can you define the relationship between organisms, right? You need to extract the 16S. This is the basic thing, which is the signature that you can look for in the DNA sequence and can compare them to identify the evolution of the genome sequence that you have. And for the first thing, 16S, we choose the neighbor joining tree. The things is that here in the the RBio tools that we want to make something very quick, and you don't need to wait for a long time in order to analyze something. That's why we choose neighbor joining tree, which is the one method that can help you identify the relationship of the organisms that you have from the 16S RNA that you extract from the genome. It is very fast because this is the way that you just create all the distance matrix, and then just reduce you can draw you can resolve the tree very fast, and then you get the result very fast as well. But there's some disadvantage, but I think we accept that other methodology can be developed in other release of the program, but this is just allow you to extract 16S RNA from the genome sequence that you interest, and then draw the quick tree to see so what is the relationship among all those genomes that you're interested. And this is the steps that you need to do in order to make the 16S RNA dendrogram here using neighbor joining. You can see on the left-hand side, there's a lot of steps here, right? Start from you need to prepare working directory, you need to create a list of genome, download genome, and everything here. And you need to extract 16S, create distance matrix, and then use the neighbor joining method to create the dendrogram here. But in RBio tools, instead of doing this is a step you need to do in the CMG bio tools. For the new tools here, you just need to prepare the list of genome that you're interested, and then you just use one command. It's called dendrogram 16S, and that's all we require you to do in order to generate this tree. So it eliminates all the steps that you need to prepare. For example, you need to prepare 16S RNA and create this matrix from this data. So you don't need to do that. So the tool will take care of it. So just only one command here, you can get this 16S RNA dendrogram here from bio neighbor joining algorithm. And this is the first thing. The next thing that this one we is currently developing, which is the genome atlas. This one will tell you this one I have just shown you that, okay, this plot will tell you what is the property of the genomes, and it requires some work as well. And always on the left hand side, you can see the step that you need to do if you use CMG by 2 or the previous tool that we released 10 years ago. And start from, the first thing, prepare working directory, pick the genome that you're interested, right? So this is one genome, right? So you need to download them and extract all the information yourself using the script, right? Do the genes, where is the gene, where is the RNA in this, and calculate all the, GC skew, position preference, stacking energy, all the property you need to calculate using the script, and then call the script to make the plot. But for the new design in R Studio here, we reduce to just only one command, which is create atlas. And again, what you need to do is tell the program what is the accession number of the genome that you want to make the genome atlas, right? You just get this accession number from the GenBank, and you don't need to download any genome. You just use this command, and then you just put the accession number to the feed in the create atlas function here, and then you get this genome atlas for that genome. That's very straightforward. One step only. And another thing that you can do is that you can plot the codon usage bias here. So what is the codon usage bias? So the easy explanation here is that, this is the difference in the frequency of occurrence of synonymous codon encoding DNA. To be easy, here is that the assumption is that if the genome have some evolutionary relationship, they probably have the same pattern of using the codon to make the amino acid sequence. And you can see from the right hand side here, as you know, one amino acid sequence, you can use multiple, it can be made with multiple DNA sequence, right? For example, the alanine here, you can use, there are 4 codes of codon here that on the DNA that you can make alanine, right? You can, the code is the GCG or the ACG or CCG and TCG here. These 4 code, whenever you see in the DNA sequence, it means that you can make alanine from that, right? So the preference, each of the genome have their own preference way to make this alanine. And you can see here from the right hand side here, the B panel here, for example, cysteine here. Most of the genome, they use TCG, TGC here to make cysteine here, except the Saccharomyces cerevisiae here. You can see there's a minus here. They prefer to use the TGT or UGU here in order to make cysteine. So from this relationship, you can make the dendrogram, or you can group the genome sequence, group the organism by the way that they prefer to make the amino acid, right? This is the way that we know immediately from the genome sequence that we have, and it requires a lot of steps to calculate this, right? You can see from the right hand side, a lot of steps, right? If you use the CMG bio tools, you need to do it by yourself, like call the gene, call the protein, and use the script to sum the codon across the genes and count the frequency, right? Just like this, and make the metrics like this, like, okay, we organism use any what kind of codon to make the amino acid, right? And then make the plot. But for the RBio tools, I don't know why I always lost my mouse. Cannot see here. For the RBio tools, you just use the one command, which is plot heat map codon, and that's it. You just feed the list of the genome that you interest, and that's all we need from you. Okay, so and the next thing is the Blastomastic, which is the last thing here that we can use RBio to compare the genomes that you interest. And before we go to the another way to compare the genome, these are all the way that you can compare the genomes, right? And another concept I want to introduce here is the pan-core and accessory proteome. So you know the genome make protein, right? And the number of protein, the different types of protein in the genome define the potential, the ability of the genome. So you can compare the protein that they make, they can make in the genome as well, in order to group them, the organism, right? That's why there's a concept called pan-core and accessory proteome. Start from the pan proteome. This is the entire protein set of all set of genomes, meaning that like in the Venn diagram, this is a union. What is the overall potential of the protein that this group can make, right? This is everything. We call it pan proteome. And the next concept is core proteome. This is, if you compare to the Venn diagram, this is the intersection part, which is the protein present in all set of genome, right? So you have pan, which is a union, core is the intersection, right? What is the protein in common that organism in your group of interest can make? This is a proteome, core proteome. And the last one is accessory proteome, which is the protein that present not in all set genome, maybe just 2 or more, but not all, right? So you have these three concepts here, and we can use this relationship to define and group organism based on the protein that they can make. All right. So, and the reason that I bring this up because this means a lot of work to be done, right? Because one organism can make, like, for example, E. coli probably can make 4,000 proteins, right, or 3,000 proteins, right? And that one just only one genome. If you have 10 genomes of E. coli, meaning that you have 40,000 proteins to compare, right? How can you easily compare this? Of course, you can do BLAST, right? But you need to do it for 40,000 by 40,000. We need to compare one by one, right? And write those relationship down, the distance, the similarity of each protein. So this one, we decide to use the speed things up, right? If you have 40,000 by 40,000, mean that you have like million times comparison of the protein. We decide to use the method called LinClust, which is the clustering huge protein sequence set in linear time. This is very fast protein clustering tools or nucleotide clustering tools, which is using the K-mer. and it a bit different from the BLAST, which is the you need to care about every single position in the sequence, right? But for K-mer, they just you need to define the window size, and then they just do the profiles from those pieces of the sequence, and then compare the query sequence with the profile you just created, right? And you can see here the way of the K-mer is very fascinated. Like you have the database genomes, right? You know that in this case, the way that they call K-mer, because this is just the how many sequence of the character that you want to divide. In this case, they choose k equal 3, right? K-mer equal 3. So you can see from the database here, suppose that you have one sequence here, and you can see if you can divide this sequence into different chunks, and each chunk contain only three sequences here, and you can see AAC, right? You can I don't know why my mouse here always lost. I cannot but anyway, it's fine. So you can see here from dividing the genome sequence like this, you can in the database and in the blue box here, you can define the relationship. You can create the tables here like ACC, right? How many times you can find ACC in this sequence. It turns out that in position 0 and position window 18 here, you have 2 AAC, right? So in the table, you can generate the, okay, how many times we found AAC at the position at the window number 0 and window number 18, and so on, right? And you do the same thing for the query sequence or the sequence that you want to search, if this sequence similar to the one in database or not. You divide in the same K, right? The same you divide in by the 3 sequences here, and you can see there's only 2 parts of the sequence here that match with the in database. You can see after you divide the sequence into 3 sequences here, only ACA and GAC match with the database here. So this protein might not match with the one in the database. You can see there's only 2 sequence here support that. Okay, how many of them match it? So if it match, like you can set the cutoff, right? If it match for some certain level of the sequence here, okay, you accept that this is probably this genome sequence, this sequence that you search probably match with the one in the database. So this one very make it very fast in terms of the comparison, the query sequence with the database sequence, because of course when you put in there, you can index them, and then whenever you have the genome sequence here, you want to match it, you just divide it into a small chunk, and then you just match it, right? And but actually, if you want to make it fast, right, you need to have a lot of the graph algorithm and everything make it works here, and everything is very written in this tool. So that's why we pick this tool to be in the heart of the way that we can define the relationship in the proteome, right? And you can see from the right hand side that a bunch of the steps that allow us to compare the proteome within the interested genomes, right? And then in the, let me see here. So in the in our RBio tools, we implement everything, so you don't need to do a lot of work in the step. You can see from the right hand side. There's only sorry. This is the step. You can see from here, there's a bunch of steps in the link trust, right? So this is what program did. So a lot of things going on. You need to create all the tables. You need to create the link, define the graph relationship, and then use the algorithm to just remove the link below the cutoff, and then you have the cluster of the proteins, right? In RBio tools, from the left hand side here is the way that you can create the relationship here, and then make the plot. From the right hand side, you can see there's a matrix, right? It means that how much the genome in terms of protein, right? How many proteome that they are similar to others, right? So for example, the E. coli K12 here on the left hand side, how much of the proteome compared to the E. coli EDL933, right? Only 64.26% here. So the tools, we call this Blastomastic, right? It's just metrics to tell you the value in size, how much similarity in terms of proteome that these two sequence, two genomes share between each other. And it requires some step and some data preparation in CMG tools. The RBio tool reduce the work that you just run in class, and you can generate these two plot by using these two command: plot blast metric, plot pan-core. You can see from the bottom, this is the pan-core plot. It tells you that when you add more genomes, you can see at first genome you didn't compare anything, right? Everything at the start point, but then you start to compare. You add the second genomes here. You can see the property change, right? The core of course going lower, because when you have two sequence, the intersection part of the protein will be lower or equal, right? It cannot be higher, right? But for the pan genome, the total in the blue one is always go up or equal, because this is every unique protein that you have in the set, right? It's a union, right? So you can just run one command. It's called plot pan-core to generate this plot. So we reduce a lot of work here from changing from the virtual machine to the R environment using R Studio here. And I think that's all. So overall, I think this is the tool that help you to do to compare genomes that you're interested. We changed from CMG bio tool, which is the virtual machine version of the comparative genomic tools, to R bio tools. It will achieve the faster way and reduce the time in comparison, and also reduce the complication of the step that you need to generate the data in order to feed in the tools that can generate the plot. So this is the all the reason why we need to make these RBio tools and what is the feature that we can have. So I will go to the demo here first. But before I move into the demo, is there any questions about these RBio tools?
Hi, I noticed whenever you use R, so is this just a giant R script that you can get exactly from like bio, how do I say, the biocomputer for the most part, or no?
Sorry, Andrew, I cannot hear you correctly. So can you repeat again? Okay. So, all right. So, if you're familiar with R, I think this one we will see in the demos that how easy that we can use. What you prepare is just the accession number in GenBank to tell you what is the name of the sequence, and that's all that you need to feed in the program, and it will download genomes and everything, do everything, compare the genome for you, and you will get the plot. Yeah. So that's very easy step. Okay, so I think we can move on to the demo. I have a little bit time left here. So okay, so start. I can share my screen. So this is my R Studio. For those who not familiar with this, or you don't use the R programming language before, you might be frightened by this, but actually there's nothing here. It's just the tools that help you to run the R script in your machine. And in the afternoon, our assistant will help you to install this. It requires some step, but after, if you can install this, is after that, doing the comparative genomic in your machine is very easy. Just like I told you before, there's a bunch of the tools that you need to install before you install the RBio tools. So this is the R Studio here, the platform that we use to run our RBio tools here. The way that you can, I will provide the folder that contain everything here you can download. So start from you need to install all the dependency. What you can do, you just click one by one, like click run here. After you after you open the folder, inside the folder that I prepare, there's 3 files here. You just install one by one, like install R. You just click here and then it will appear on the left hand side here in the code area here. You just click each of the line and then click run. And you can see instantly from the left, the lower left here, see after you click run, it will take the package from internet here and then install it in your machine. So you can just click one by one, right? For example, here, another package called FMSB. See, after, if you successfully install, you will see it on the left-hand side here. You can say, it will say that, oh, it installed here, then download in this folder or something. So you just do it one by one. So this is how, very easy, and you don't need to switch between windows, right? If you have a lot of programs, this is just only one place. Everything will be done in one place in R. Right? And the goal of the first day of the workshop is to install all of this. So after you install everything here, but actually you don't need to click one by one like this. If you install R in your machine and RStudio like this, you can click at the package here on the right hand side here as well, and click install here on the left here. And you can select the repository CRAN. And if you want to, for example, you want to install the P heat map here on the left hand side here, you just type the name, right? If you don't want to use the command line, you can just use the tools built in RBio tools, like P heat map. See that after you type some word, it will look for this package in the repository in CRAN here, and then, oh, if it's available, you just, okay, you want to install, just click here and then click install, and that's all you can do here, right? You can either use our the script that I provide here. You can click one by one here, and then you just finish all of this, right? P heat map and R curves like this, and you can see it will download and install automatically in your computer one by one here. So I prefer to do it one by one, so you know the error. If there's some error, something happened in your machine, you will know from the lower left here. And this is a crucial step in order to install the RBio tools. Then BioC Manager here. See all of this available in your computer now. You can check in the, you can see in the right hand side, there's the pane called package here. You can check if you install successfully or not. If you install it successfully in your machine, you can search it in the package search here. For example, the package called P heat map, right? Yeah. If you type here, you can see, oh, this one already installed. You can see the name appear in the pane here, which is the name of the package installed in your computer. Right. This is very easy. And then this dependency from bioconductor, you just click it like this. Just click at the script, and then, and sometimes it will ask you, oh, this biostring, the package here require, there's a newer version of the dependency you want to update all. But I sometimes you don't need to update all of them because it might not work, right? With the dependency, the rest for RBio, you just say no, n, right? Just type n in the command here. I don't want to update, and just that's it, and everything. And for example, find my friend here, the program in the bioconductor, you just click in one by one, and if it asks you, oh, you want to update, of course, I don't want to update right now. Just click no, and that's all. It will install in your computer without update all the dependency, right? Sometimes it after you update it, it might not work, right, with the rest of the software. Just okay, I don't want to update right now, and that's it. After you install all of this, the next is to install the RBio tool itself, which is the local package, right? So you need to change from here, install using the install method in the bottom right here, pane here. You just click install and then change to package, archive files, and then it will tell you, oh, where's the package. So I put it in here. It's in BioTools in the local packages. I will ship all of this to you. It's in local package. The first thing that we need to install is the protocol here. You click protocols and click install, and it will compile. That's why you need to install C compiler in your program first, in your machine first. For Windows, it's RTools. For Mac, it's Xcode. You need to install it first in this afternoon, and then you can begin all of this. Right now, it used the C compiler to compile all these local package to your local machine. It will take a little bit of time, but hopefully it's not that long. So here it will compile. So in total, we have 3 different local package here. Oh, I cannot see. Okay, why it's running. All right. So if it's successfully installed, you will see it will tell you, oh, done, and then protocol successfully installed in your computer. And in total, we have 3 different local package, right? Here, install protocol first, install RNAMer, another one, and the last thing is RBio tools. And this shouldn't take too long. And it's just one time, right? You install and that's all. You can use these tool anytime to analyze for the genome that you interest, right? So you can see RNAMer done here. You can always check if you install successfully by typing here, do you have RNAMer? And you can see you appear here. Okay, this worked. It was installed in your computer. And the last step is the RBio tool itself. You just click this, the local machine in the tar format here, and then click install. And this is the last step of the installation part that we will do it for today in the afternoon. Yeah. The reason that it might take the whole afternoon because sometimes you might need to wait to install RTools, and install Studio in your local machine. And for those who use Mac, you need to install the Xcode, which is the tools that allow you to use the C compiler and everything in your Mac environment. So it might take like 1 hour of installation, and it will download everything from internet. So it might take an hour. Right? So now, after we install all the dependency here, we install the local package one by one. At the end, we install the RBio tool already finished, and you can always check from the package pane here. You can type RBio tool, and you can see it's here, RBio tool. And in the Mac, in the no, I don't want to. Okay, so and it's shipped with the manual. So the way that you if after you install it, you can always do the can see the question marks here, and then if you want to see the description and the manual of the package that you install in your machine, you can do in the pane here, in the console here. You just do the question mark and then followed by the package that they want to you that you want to see the description, like in this case RBio tools, right? And you click enter, No documentation. You need to load it first. Just click here. You can click by tools. Oh yeah, you can just. Let's see here. Library. By tool. Like this and run. First. Oh, okay. Now it's. Can try again. And you can see on the right hand side here in the pane. So this pane, the bottom right pane, it combines everything. If you have the plot, it will show in the plot pane here after you run some script. If you have the pack, you can check the package and you can check the help here as well. And you can see, and this is the description and the feature that we have. You can see there's a lot of feature that you can use by to analyze, quickly analyze the genome that you interest. Right. I'll give you one example here, and you can read it after you install it. So, but the first goal for the first day is that install this thing in your local machine, and that's all. And in the folder that I will ship you, there's a, it's called tutorial 01 here. Just want to tell you, okay. For example, we have the this is the way that you can run the tools. First thing you need to load the library first, and then you initialize the object, the global variation here. You can see this is the like an empty folder in the memory that RBio will work on the data. And then you just select the genome sequence that you want to analyze. And the code here is the accession number for each of the genome in the database. In this case, this one, I got it from GenBank, which is the public repository that contain all the genome. And I want to use this one as the set of genome that I want to that I want to analyze. Just run it. And you can see here, there's this set of accession number will be stored in this variable. Right? You can see all this contain all the name here. And if you want to make the dendrogram, just like I told you, the 16S RNA from bio NJ, you just click just one command, and that's all. And you feed in this command with the set of the genome that you just create here, set up the name here. It will download extract 16S and then do the plot for you. You can see here after I run this command, click here and then click run here. You can see from the console pane, it will do everything for you, download the genome, the first genome, and extract the RNA gene for you. And everything will be in the memory. And you can see it will appear on the right-hand side, up right here. This is the empty folder for the RBio to work on the genome that you interest. And you can see it will download and extract 16S one by one. In this case, we have 6 genomes here. It will download one genome at a time and then extract 16S, all the RNA for each of the genome that they after they download the genome. And you can see here one by one here. So we have 6 genome. It will take a little bit of time, but believe me, comparing to the previous version of the BioTools that we have, which is the CMG BioTool, this one is much faster and then very straightforward. And compared to the the previous version, you need to extract by yourself using one command at a time, and then you need to tell the program to make the tree. But for this environment here, using BioTools here, just one command. What you need to have is just the accession number of the genome on GenBank, and that's all that we need from you. The rest, the program will take care of it. You can see here now we are at the genome number 5, and then this is the last genome that they will download and then extract 16S. After that, it will compare all 16S. And you can see now it's done. You don't need to use a lot of the script to generate the 16S and a dendrogram here using the neighbor joining algorithm here. So now it download everything for you. And you can see the leaves here is the corresponding to the strain name here. And you can see this is the relationship in terms after we extract 16S RNA from this 6 genome, and this is the results will be appear on the right hand side in the export in the plot pane here. And sometimes it will if the script cannot be seen here, it will generate the files within the environment that you are on here. You can just click and view it. And that's all. I think this will show you how easily that you can make the do the comparison of the genome, just one click and one command using the RBio tools. All right, so that's probably it. And this is the useful link that we will do in the afternoon, I think, here. Okay, you probably can see here. So this is the link for all the software, the RBio tools and the steps. Everything will be in here. So you just follow the link, and then you will see the path.
Visnu, can you paste that into the chat? And that way all of the participants can click on it.
Sure. Where's the, all right, in the chat box. I need to stop sharing here. And all right, here, this is the everyone. All right, this is the link to the path, the online path that I put all the details. How can you get the software and the steps of the installation? So this link we'll use in the afternoon for you to install and download the program that we have today. And share this. Okay, this will link to this kind of this is the path online. After you follow the link, you will see here. This is the link to download the R programming first, and then you need to have for those who use Windows, after you download the R, you need let me do this, maybe easier. This. And then you need to install the C compiler in your program after you have R. You need to, for those who use Windows, need to install the R tools first. But for those who use Mac, need to install the Xcode. So, and this is the YouTube link. I think this is a very useful link. It will guide you how, for those who use Windows, kind of complicated, a little bit complicated, but not a lot, to make the R and R tools work in your machine. And that's it. And after that, there's a link to download our the RStudio, that the one that I just have shown you. And then at the end, after you install all of this, you are ready to install RBio tools. And this is the link to the software, the folder on the box. And after you finish all the installation here, after you click here, this one, it will lead you to the box that we have. For the software, there's two files here and one folder, the local, and you can see here, this is the local package that I just showed you how to install them. And that's all. Everything will be in this the path online.